I'd like to introduce today's speaker. Today we have Dr. Monica Cooper. She's a viticulture advisor with UC Ag and Natural Resources and UC Cooperative Extension in Napa County. And today she's going to be speaking on in-house diagnostics for disease management, LAMP for red blotch detection in grapevines. So you can go ahead and share your slides.
Okay, great. Thank you, everyone. Thank you for being here, and thank you to Cheryl and the UC IPM program for the invitation. So, as mentioned, I am going to be talking today about the detection of grapevine red blotch virus using the LAMP assay. So that's a project that my team and I have been working on since 2021. And so I'm really pleased to be able to talk about the assay itself and then how we've been using it in vineyards. Okay. So to give you a little bit of context, today we're going to be talking about the grapevine red blotch disease system. So the pathogen that we're talking about is grapevine red blotch virus. It's a double-stranded DNA virus that infects grapevines. It was first identified in California in 2012, but we know from herbarium specimens and sort of other anecdotal observations that it has a long history in California. And in fact, there's even the suggestion that this virus evolved in California from a latent virus that is found in our native grapes, Vitis californica. So this virus infects plants in the Vitaceae family. So that includes Vitis vinifera, so our cultivated grape varieties, as well as the rootstocks that we use. So much of that is American parentage, and as well as hybrids. So this is, going to affect grapes regardless of whether they're destined to be table grapes, juice grapes, raisins, or wine grapes. We've tested over the years a lot of different cover crops that are used in vineyards, as well as weeds and some adjacent sort of natural vegetation. And what we found is that the virus does not infect and cause disease in any of these plants. So really the host range of the virus is limited to plants in the Vitaceae family. So there's two ways that the virus can move around. One is in plant material. Given that grapes are asexually propagated, if you take your budwood from a vine that is infected with grapevine red blotch virus, and then you graft those buds onto your rootstock, or if you're top grafting, you can move red blotch from one plant to another via that sort of mechanical way of grafting. But the other thing that we see is secondary spread with the virus. And that means that the virus can move via an insect vector from one grapevine to another. And the only insect vector that has been identified to date is Spicistolus festinus, which is the three-cornered alfalfa hopper. And that is pictured here. It's kind of a cute little guy if you're into insects. And it, as the name suggests, is a pest of legumes, principally in the southeast, but also in California. So it's a known pest of alfalfa, peanuts, and soybeans, and it has in California and other places, it has a very large feeding host range, although its breeding hosts are limited to those plants that are closely related to alfalfa, so legumes such as clovers and vetches. And the reason why we care about red blotch disease is that it has physiological impacts to the vine. So this is a phloem-limited virus, and it's going to affect photosynthesis and stomatal conductance. And then the sort of downstream implications of that are that the sugars that are produced via photosynthesis get stuck in the leaves. So they accumulate in the leaves and they're not transported into the fruit. And so if you're trying to ripen your fruit, there are very clear impacts on ripening. So sugars and anthocyanins and other aroma compounds do not accumulate in the fruit until you end up with fruit that doesn't have a lot of great flavor or color. And it also is very low in sugar content. In some cases, those clusters will stall out, or the berry ripening will sort of stop altogether at a sugar content that is really not meeting any contract obligations or meeting the standards that it might need to make in order to be turned into a approachable wine. So what we see is that the wines that are made from fruit coming from red blotch vines have less body and aftertaste, sort of low in color and fruit character. They can be quite acidic and sort of vegetal in character. So this does have severe sort of economic impacts. The visual symptoms of red blotch disease, just like the name suggests, are blotchy, irregular spots on the leaf blades. And those symptoms are most obvious from around the time the grapes start ripening, but mainly into the fall. So in Northern California, we're looking at symptoms in the months of sort of September and October. Usually we're looking at symptoms between mid-September, and then we stop by Halloween because the leaves are senescing and we don't see symptoms very well anymore. This disease is incurable, and so that means that the only way to lessen the inoculum that's either in your vineyard or in a neighboring vineyard is to actually remove those vines. So we need to go through and determine where the vines are located, mark them in some way, and then come back and actually remove them. And that can either be done on a vine by vine basis, or when you get to a certain proportion of the vines within a block that are diseased, you may have to remove and redevelop the entire block. And so a lot of the mapping efforts are done just by people either walking or driving slowly in ATVs, recording the presence of symptoms. Although there's also been a fair amount of work on proximal and remote sensing for this disease. And so it's possible that in the future it will be a little bit more automated, which could make the process of cataloging where the vines are more efficient. As I mentioned, mapping is principally based on visual symptom identification, and visual symptoms can be a little bit tricky. So it definitely takes time to get an expertise to be able to pick out, oh, that's a red blotch diseased vine as compared to a vine that might be expressing symptoms of a nutrient deficiency or another disease or pest. And on top of that, the red blotch disease itself, the symptoms can vary by location, season, and cultivar. So some seasons, the symptoms are quite clear. It's really easy to pick out which vines are diseased. And then other years, just because of environmental conditions within that growing season, the symptoms can be really mild or very difficult to distinguish between those symptoms and, say, a nutrient deficiency. So there's a fair amount of expertise that goes into being able to recognize these symptoms, and with experience, people get much better at being able to distinguish the symptoms of red blotch disease from another malady. The other factor that plays into this is that there are some cultivars, such as Sauvignon Blanc, which do not ever really develop symptoms. So they can be infected with a disease, and you just never see any visible manifestation of that on the leaves. And if you're interested in reading any more about that, we have an article published in the American Journal of Enology and Viticulture that sort of walks through the steps of monitoring for visual symptoms and some things that are you might do or not do if you're going down that path to do some symptom mapping. Okay. So back to the content, because visual assessment takes time and expertise, a lot of times we want to make sure that our visual assessments are accurate. And we do that with diagnostic assays. PCR is a sort of standard of what's been used in historically. And so you're collecting a sample from the grapevine that you're interested in its virus status. You're sending those samples off to a diagnostic lab. And then, within a few days to a week, they send you a result back telling you which viruses are found in your sample. What we started doing with this project is actually doing an in-house assay called the LAMP assay for red blotch virus. So that's loop mediated isothermal amplification, which is a point of use benchtop assay for red blotch. And the great thing about this assay is that you can do it in-house. So you collect your sample and then you go through all of the process of processing the sample and running the assay. And then you get a result without sending your sample off to a diagnostic assay. So what LAMP does is that it detects and amplifies viral DNA from red blotch infected vines. That amplification, because of the master mix that you add to each of the sample tubes, if there is virus in that tube or in that sample, there's a color change. So pink means negative and yellow means positive. And as I mentioned, one of the great benefits that we found with this, because I also don't have access to a diagnostic lab, is that we can do this sort of in our office in a clean space on a bench top without a lot of fancy equipment or really too many reagents either. And so here is the process of LAMP for red blotch virus. So the first thing you're going to do is you will be collecting plant material. As if you were sending it for PCR, that plant material can be petiole material or it can be canes. Another thing that we found through the process of working with LAMP for red blotch is that we could actually collect our sample material from trunks. And that is an innovation that we made as part of this project. And you're seeing that here in the video where this is a vine that's been received from the nursery, and Hannah is going about the process of collecting tissue for analysis. So once you have your sample, you peel back to, if it's a cane sample or a trunk sample, you're going to peel back to get to the cambial tissue. And then you're going to use a pipette tip in order to collect the sample tissue that you're going to use for the analysis. So there you can see in the video where she is doing the collection. And then these are the still images here showing you the tissue in the tip of the pipette that is then put into another tube that has distilled water at the bottom of it. And that becomes the template, which is what is used for the assay itself. So that's the process. You go to your grapevine that you're interested in, you collect your plant material, you sample that plant material with a pipette tip, and then you create your template that's going to be used in the assay. So when you're going to do, which tissue are you going to use and when? And what we found is that we can use petiole tissue for the LAMP assay between veraison, which is the onset of ripening, and just about pre-senescence. So once those leaves really start turning yellow, we no longer use the petioles, but that during that period of time, sort of in the ripening period of time, through harvest, we can use petiole tissue. Once the petioles are no longer suitable, we move over to using canes. And we use canes basically from that sort of pre-senescence period through to when they're pruned off. If you're enterprising about it, you could actually do all your sample collection of your canes and store them frozen. So before you go through that step of collecting the tissue from the canes, you can actually store them frozen. And so that can extend the period in which you can use the canes for sampling because you can store them in the freezer. Once those canes are pruned off, getting ready for the next growing season, then we start using the trunk tissue. So between pruning and the onset of ripening is when we use trunk tissue because that's the most reliable tissue for the assay at that period of time. The neat thing about trunk tissue that we found is you can actually use it year round. So if you like using petioles or you like testing canes, that's great. But if you get into a rhythm of testing or you're really familiar with trunks, then you can actually use them year round. And so here's a little bit more detail on how you might go about collecting trunk samples. So basically, first you're going to peel away all the outer bark, and then you're going to come in with a clean razor blade and collect the, sorry, and then cut down in order to get to the vascular tissue. You're going to grab the pipette tip and use it to collect the sample from the vascular tissue. As you can see here in the photograph where there's a tissue in the tip of the pipette. And then you're going to put that while you're still in the vineyard, you're going to take that pipette tip and place it in a tube with distilled water. And then that becomes your sample template that you take back to the office or to your lab space and use to run the assay. And what we found over time is that location matters when you're collecting these samples. So red blotch virus is unevenly distributed within grapevines. And what we've seen in our studies and in other studies conducted in Oregon is that the basal tissue or the older tissue is more reliable. So you won't get false negatives if you're using the basal tissue. So we really focus in that sort of lower canopy when we're collecting petioles and then also collecting canes, of course, respecting whatever wood you're going to need for the next growing season. So we focus on a bilateral cordon, on collecting samples across the cordon. And then if you had a quadrilateral trained cordon, you want to make sure you're on both sides of the vine that you're collecting tissue. And so what we've seen is that if we are only collecting tissue from one side of the vine, particularly in quad trained vines, we can get really mixed results. So a sample from the northeast side of the cordon position might come back positive, whereas one from the southwest may come back negative. And so that's really why it's important to make sure that you're collecting from multiple locations and that you're representing the vine adequately when you're doing your sample collection. As I mentioned earlier with the canes, they can be frozen for up to a month before you process them. So that's a nice way of sort of, like I said, extending the season that you can use those canes. The petioles, we don't freeze those, but you can refrigerate them for up to a week before you go through the sample collection process with them. And then if you have your templates, you can store those in the freezer for 3 to 4 months before you test them. The reason why we don't store the templates longer is that we see evaporation even in the freezer in a closed tube of that water in the bottom of the tube. And so we've found that within 3 to 4 months, you really need to be testing those so that you make sure that you have enough sample to test. And there is some sensitivity to how much tissue should be collected. So if you collect too much tissue, you can get a false positive. And that happens because of the pH change in the reagent, which can cause a color change even though there's no red blotch present. And so the tissue that's most sensitive to that are the petiole tissues. So when you're doing that petiole collection, you really need to be conscious that you're not putting more than 2 millimeters of tissue in the pipette tip. Whereas the canes are a little bit more forgiving. So you can do less than 2 millimeters up to 5 millimeters and get really reliable results. Once you get more than 5 millimeters, you're going to start getting unreliable results with the canes. And the trunks are the most forgiving tissue. And so in addition to the fact that you can test, from trunks year round, they're also favorable because they are forgiving in this way, and you don't get a lot of false positives based on having, too much tissue in your pipette tip. Okay, so once you have your template ready, you've collected your samples, you're going to be ready to conduct the assay. So that means that you're going to mix your reagents and dispense them into the PCR tube. So that includes your primers, your distilled water, and your master mix, which is used to elicit the color change. After you have your reagents in your PCR tubes, you will then take the templates, which are your samples, and add those to each of the individual tubes. And you'll also add your controls at that point. After that, you will close all those tubes very tightly and incubate them in a heat block for 35 minutes. And during that time is when the amplification is happening, and that will result in the color change. So once you pull them out of the heat block, anything that has a yellow color in the tube means it's a positive, and all of our samples, which are numbered 1 through 4, did not have the virus present in them, and so the master mix did not change color, it stayed pink. And it's really important here that all of our negative controls also stayed pink. And for sample setup, when you get started, you'll have 8 PCR tubes in each strip. And we recommend that you start with 4 samples. So these are the vines that you're testing, and then that you also use 4 controls. So you'll have a positive control and 3 negative controls. And each of those negative controls controls for a different step of the assay. And as I mentioned, if one of those negative controls turns yellow, it means that you have contamination somewhere along the way and you can't trust your results, so you need to go back to the drawing board. The good news is that as you gain experience and proficiency, you can reduce the number of controls, which will allow you to increase the number of vines that you're sampling. So one question that we get often is who can learn how to use LAMP? And in our experience, anyone can. So there's certain key skills that are really important, but this assay, it just takes a lot of practice and a lot of attention to detail in order to gain mastery. So key skills include organization. So you need to be able to label and track your samples. You need to make sure that you know that you put all the reagents in the appropriate tubes and added your samples, to be labeling things along the way. It helps if you have laboratory experience, but we found that it's really not required, although you do have to be dedicated and diligent about being clean. So there is a high risk for contamination. Once you have the DNA somewhere that causes contamination, it can be really difficult to clean that up. And so you really want to make sure that you're keeping your space clean, that you're cleaning your hands and your equipment and your tabletop, and make sure that you're keeping any samples, in a place that's not contaminating where you're running the assay. We've also found that sometimes things do go wrong, and so you need to be able to think on your feet a bit and solve any problems that arise. And there is a learning curve, so we've taught a lot of people how to use the LAMP assay over the years, but sample collection and processing quickly become routine. So once you've done this, 100 times, maybe 1,000 times, you get quite good at it. So there are some things to learn around sampling, particularly, as I mentioned, you really have to make sure that you get the right amount of material from the plant in the pipette tip. There are multiple steps within the assay, and so you have to sort of develop what your workflow is going to be and how that works for where you are in your workspace. As I mentioned previously, always, always, always be clean. And then you need to, if you haven't used some of these laboratory equipment before, like pipette tips, then you want to get comfortable with using that equipment. Okay, so in order to reduce the risk of contamination, we recommend that you clean your workspace, equipment frequently with bleach wipes or a bleach solution. We wear gloves and we change them between steps. If we're not wearing gloves, we're washing our hands frequently. We're taking our reagents and aliquoting them into smaller portions, and that makes it easier so that if you do have contamination, you don't lose a large portion of your reagent, you only lose that small aliquot. And as I mentioned, we also keep anything that we use out in the field for collecting the samples, etc., away from the area where we run the assay. If you do get contamination, you need to clean thoroughly and then do some testing with controls only, and so that you can figure out where that contamination is coming from. Did you accidentally contaminate the water or one of the reagents, or is that contamination coming from somewhere else? Okay. For the last part of the talk, I'm going to talk about how the LAMP assay is being used in practice. So, as I mentioned, we've been doing this in-house at UCCE Napa, but we have also been working with many growers in Napa and broader North Coast region who have started using LAMP. And so, as I've already mentioned, one of the ways that we use LAMP is to complement and confirm our visual assessments. So we go out in the vineyard in the morning, we look at some vines, and we decide whether or not we think that they have red blotch symptoms. If we're questioning our visual diagnosis, we can pull samples from those vines, bring them back to the lab, process them, and determine whether or not they are infected with the virus or not. And so what we've seen over the years is that LAMP has really good correlation with our visual symptom assessment. So vines that we mark as symptomatic tend to be infected with red blotch, and vines that we mark as asymptomatic tend to not be infected with red blotch. So we did have a couple vines that were not symptomatic, but they were infected, and that's not uncommon. Just they were infected, but they didn't have visual symptoms yet, and we don't get a lot of vines that are symptomatic but do not test positive. So it's really great that we have pretty good alignment as far as that goes. We've also been using LAMP when we don't have visually diagnostic symptoms. So as I mentioned earlier, there are some wine grape cultivars like Sauvignon Blanc, which never develop visual symptoms. They can be infected with the virus for years, and you can never see any change in their foliage. And then in other cases, such as in the example photos here, B and C, we have co-infections with leaf roll virus. And in that case, we see the symptoms of leaf roll virus, but we can't also tell that those vines have red blotch. So we started using LAMP in these cases because we knew that in some cases, the Sauvignon Blanc blocks were serving as a source of inoculum because the adjacent Cabernet Sauvignon or other red wine cultivars were turning red. And so we knew that the virus was probably coming from some of these old Sauvignon Blanc blocks. And the process that we use in these instances is called adaptive cluster sampling, which is a statistically driven approach to identify at-risk vines in the vicinity of known infected vines. So what we know about red blotch is that we get a very clumped distribution. So you'll have patches of vines where multiple neighbors have the disease. And so this adaptive cluster sampling is a way to organize your sampling such that you do first a random pass, and then you go back in and sample near positive vines, because you're looking for places where there are clusters of infected vines. And we did that for our research trials in 3 blocks, one in Oak Knoll, one in Rutherford, and one in St. Helena. The Oak Knoll and the Rutherford blocks were Sauvignon Blanc, and the St. Helena block was co-inflected with lethal 3. And the first thing we did was divide the block into equal sections. So in our experimental blocks, we did that some using based on sort of where our traps were located, but you could do that in a very grid type pattern. So you take the block that you're interested in and divide it into equal sections that are between 0.2 and half an acre in size. And within each section, you're going to randomly select 8 vines, and you're going to conduct your LAMP assay on those sample vines. If you don't have access to LAMP, you could also do this using sort of traditional PCR methods. Collect samples from those vines, send them off to a lab, and determine their red blotch status. So that's the first step. That's the random pass, where you are taking your entire population and you're sampling from it. And what you're hoping to do is then find these pink or red cells that you're seeing here, which indicate vines that are infected with red blotch virus. So the blue vines are vines that were sampled but did not have red blotch, and then the pink or red vines are the ones that were sampled and did come back positive for red blotch. Once you find those vines, you're going to go into the adaptive approach. And so the next step is, next to all the positive vines, you're going to sample the 4 neighboring vines. And this is the step that's going to allow you to identify where those clusters of infected vines are. And in this example, I'm showing you one step where you had the original infected vines here on the left, and then on the right, you're seeing where we sampled those 4 vines around them. When you get positives within that second sampling, you would then go out from any positives in that round and sample the adjacent neighbors. And you can repeat that as often as you need in order to determine what the spatial distribution of infected vines is within the vineyard block that you're interested in. And this is what it looks like for those blocks that I mentioned that we did our study in. So the Sauvignon Blanc blocks in Oak Knoll and Rutherford, and then the Cabernet Sauvignon block in St. Helena that had a mixed viral infection. And you're looking at 4 years of data for the Oak Knoll block, and then 3 years of data for the other 2 blocks. And what you're seeing are the results of the cluster sampling. So we're finding in this corner of the block here at the bottom that we found a cluster of diseased vines there. So we know that there are a lot of infected vines here, and by the final year, we were also finding clusters of infected vines in the middle of the block and at the other end of the block. Compared to the other Sauvignon Blanc block, where we had a very small cluster and it was fairly isolated in this one section of the vineyard, and then in the Cabernet Sauvignon block, where the infections were also somewhat isolated, although not quite as isolated as that Rutherford block. And so this approach, using the LAMP assay in these blocks that either don't have symptoms or that have very visually confusing symptoms, really allows us to get a spatial understanding of the virus and its distribution through time in these blocks. the other thing that we did was collect feedback on from the people who are using LAMP for red blotch within Napa at the time. So during the period when we were doing this study, there were about 20 or so people that we had identified, and 10 of them responded to our survey. And so I present this information to show you that not only is the assay being used, but then also to sort of show you um, the things that you might need to consider if you were going to adopt the assay. So as I said, it does take a little bit of time to get proficient in the assay. And most of the people who responded to the survey said that by the time they had done around 50 samples, they felt like they were proficient. For a few of the respondents, it took 150 samples or more before they felt proficient. But that gives you kind of an understanding of how long it takes to develop that proficiency. At the time, the sort of average cost of the supplies and labor to both collect and process the samples was between $7 and $10 per sample. Of course, that is never a fixed value given, inflation and other sort of changes in the cost of labor, supplies. But in general, it's probably maybe at this point between $8 and $12 per sample. Most people who were using the assay were using the trunk. That's where they were collecting their samples from. And we heard that not only is that because they like the flexibility of when they could collect the trunk samples, but also they felt like it was pretty easy to learn how to collect them and fairly easy to get proficient. And then, as I mentioned previously, you don't get a lot of false positives. So it's a little bit forgiving as far as tissue goes. So it was definitely the preferred tissue, and canes were second most preferred. And I think that is because that cane tissue can be stored over time in the freezer. It takes on average like 1 to 2 days to collect samples. And as I mentioned previously, most people were collecting trunk samples, and most people had between 1 and 2 people doing the collection and then processing the samples. And although we were teaching people about LAMP, people who had become proficient were also teaching each other. And so we saw that the process and the assay was being adopted because of the social networks that had formed within industry and between users, and they were sharing information among users. So I think that's a really great thing. So if you're interested in learning more, we have a lot of resources on our website, including, a very detailed protocol. We have a cost and materials handout, how to order the primers, frequently asked questions, because inevitably, even though we try to cover everything in the protocol, there are certain questions that come up. So we've made a list of the ones that we get most frequently, how to sample for trunks, how to do the adaptive cluster sampling if you are in a block that you don't have any information about the visual symptoms, and then other types of resources on how to use the LAMP assay, particularly related to if you're testing trunk tissue. And once you have all of this information, of course, there will be decisions that will need to be made about the disease and how you're going to approach management in the block that you've been working in. And so also on the website, you will see that we have handouts that go through the disease ecology and the different management based on sort of the percent incidence of symptoms, and then also whether those symptoms are aggregated or random. We also translated that into Spanish, so you will have that resource in Spanish as well. And we did analysis of the financial considerations for red blotch disease management. So as you're going about determining what you're going to do, you can also look at sort of the implications that might have from an economic perspective. So I'd like to thank you very much for your attention today. I'd also like to thank my team who has been working on this with me for the last 4 years, to the funding that we get from CDFA through the PD Gwist Board. That is grower money that we are very appreciative of. And then also to allow you to have the link to our website. And then if you wanted to do any more reading about the work that we've done with the LAMP assay for red blotch, there are 3 articles that we have published in the American Journal of Ecology and Viticulture. And that concludes the content that I have for the day. And so I'd like to open it up to see if there are any questions.
Yes, hi. Thanks, Monica. I appreciate you presenting. There are a few questions, so I'll go ahead and read those to you. The first one was, is there any guide on cultivar and symptomology?
Oh, that's a good question. Not really. So there's some of that in the in the article that I talked about that we wrote, but it doesn't really list all of the different cultivars and what the symptoms might look like. So that's something we could produce, but there's so many cultivars of grapevines that we don't have photos for all of them.
Okay, thank you. And then the next one, have you ever studied minerals in the leaf with and without red blotch symptoms?
I have not studied that specifically, but there have been other articles published around sort of vine nutrition and other impacts of the disease. So you'd have to look in the literature for that.
Okay, and then there was a question coming in. Is the razor that you used, is it a single use or is it sterilized between samples?
Oh, so it's single use, but we reuse them. So we single use them and then we come back and we sterilize them. So we wash them and then we don't have an autoclave, so we bake them. But some people put them in like a crock pot or a toaster oven or some other way in order to sterilize them. But yeah, they are single use, but then we reuse them.
Okay, and then this next one came in, are bleach wipes or solution appropriate for disinfecting equipment? It is my understanding that equipment must soak for at least a half an hour to work, and isopropanol or ethanol would be needed to be used for immediate disinfection.
We do most of our disinfecting with bleach wipes. So we do make a solution, but we do most of it with bleach wipes.
Okay, and then another question. Would you please explain about the insect vector of the disease, how to transmit, and more?
Oh, that's like a full nother 30-minute talk. So um, yeah.
We might have some information on that. I'm not sure. In the pest management guideline, I'm not completely sure of that, though. All right. So another question that came in, what is the estimate of equipment cost to get started if you have a clean area to work in?
Oh, so I don't have that off the top of my head, but if you go to the website and you look at the materials list, you can get that information. We have that. We have the latest cost. We also can't keep up with it because, it's ever changing.
Okay. And then this one's from Mohammed. Thank you for the presentation. Are there any varieties tolerant to the red spot virus, a red blotch virus of grapevine?
So yeah, as I mentioned, like Sauvignon Blanc can get infected, but it does not really have visual symptoms or fruit symptoms. But there is no like known resistance as far as we know. But there's definitely differing levels of the sort of plant response to the virus. And by that, I mean also like the degree that the virus is affecting the fruit and ripe ripening.
Okay, and then another question, what about disease, chemical and cultural controls?
So there are no chemical controls for the pathogen itself. There are insecticides that will kill the vector, but what we don't know is um, so the vector would be killed, but we actually don't know what the effect is then on spread of the disease as a result of that. So that's still ongoing work.
Okay, and then just two more questions here. And this one, do you recommend testing one side of the trunk or two sides of the trunk?
Um, yeah, that's a tough question. I think, yeah, if you could, not necessarily sides, but definitely at two different heights and maybe a little bit around the sides. Without having to move. So if you have to move from one side of the vine to the other, that's not efficient as far as time goes. So testing like in two locations on the trunk, maybe one upper, maybe one a little bit lower, kind of offset from each other, I don't think would hurt.
Okay, and then just this last question came in. What are your thoughts on the potential of other vectors? It seems like the TCAH has low population numbers.
Yes, Tika has low population numbers, but I'll remind you that so does great mealybug, and it is a quite efficient vector of leaf roll disease. So just because you're not seeing the insect doesn't mean that it's not out there in large enough numbers in order to be moving the disease around. I think this is a similar situation for Pearson's disease, where we don't catch that many blue-green sharpshooters in our traps. We don't see them usually when we're scouting, and yet it's enough to create epidemics of Pierce's disease. So, and then the second aspect of that question is, are there other vectors? So these insect uh, pathogen or relationships are long, they develop over time, right? So evolutionarily. So this virus is in the phloem. The insect has to feed in the phloem, pick it up in its mouth parts. The virus has to go into the digestive system of the insect, go back out into the insect body, and then go into the salivary glands, and then to be moved back into another grapevine. So there's a lot of points in that process where the virus could either be expelled by the insect, where it could be recognized as a foreign particle, sort of isolated within the insect. And so the likelihood that there are going to be vectors that are not within the same family of insects is very low. So, and there's really only two tree hoppers in California that occur in vineyards, and those are Tika, three-cornered alfalfa hopper, and tortoiseless. So the odds of there being another vector of importance in vineyards in California are really low. And then the next question is, what is Tika? That's three-cornered alfalfa hopper. That is the vector of grapevine red blotch virus.
All right, thank you. I think we got all of them. Just wanted to thank you again, Monica, for presenting today. Appreciate that.
Great. Thanks, everyone. Thanks, Cheryl.
Thank you.